Hello everyone, and welcome back to the series of lectures on cross-couplings. You have been introduced to cross-coupling reactions, and in the previous lecture, we compared the reactivity of the most common coupling partners used in cross-couplings. In this lecture, I will describe the factors controlling the selectivity in transformations involving substrates with more than one reactive center. Substrates possessing multiple functional groups capable of undergoing an oxidative addition are common in complex organic synthesis. For such substrates, finding conditions for the site-selective functionalization of one position over all other positions is critical. To understand how selectivity arises in the cross-coupling of polyhalogenated arones, one must first consider the mechanism of cross-couplings. A simplistic depiction of the catalytic cycle for a transition metal-catalyzed cross-coupling reveals three key elementary steps: oxidative addition, transmetallation, and reductive elimination. Oxidative addition is the step where the electrophilic coupling partner is introduced into the catalytic cycle. For most instances, oxidative addition determines the selectivity of the process. In this step, selectivity arises due to differences in the relative electrophilicities of the carbons bound to halogen groups in the parent arene. As oxidative addition formally involves the addition of electrons to a carbon-halogen bond, the most electrophilic carbon would be expected to be the preferred site of attack. Traditionally, site-selective couplings have been achieved by employing arene electrophiles bearing multiple, non-identical halogens or pseudohalogens, such as triflates. When the selectivity is determined by the use of non-identical functional groups, it is called chemoselectivity. In this case, chemoselectivity relates to the relative bond dissociation energies of carbon heteroatom bonds, which, in turn, are associated with the polarity and polarizability of the bonds. Therefore, the site selectivity in arene electrophiles bearing multiple, non-identical leaving groups follows the general reactivity trend we learned in the previous lecture. For instance, if you try to perform a Sonogashira coupling for this multihalogenated isothiazole with not particularly active catalysts, the reaction will first proceed at the carbon-iodine bonds. By applying higher temperatures or sterically hindered sigma donor ligands, one should be able to sequentially functionalize the carbon bearing a bromide. The site-selective functionalization becomes challenging when the parent arene bears identical halogen atoms, as there is little difference in the nature of the aryl halide bonds. The site-selective functionalization of systems possessing the same functional groups is otherwise called regioselective functionalization. In these systems, regioselective coupling at the carbon bearing one halogen atom over another depends on other factors, such as the electronic nature of the atom to which the halogen or pseudohalide is attached and the steric environment, which determines the preferred site of reactivity. Recent studies indicate that the regioselectivity of cross-couplings can also be controlled by the used ligands, solvents, and additives. All these factors, along with specific examples, will be discussed here. First, we will discuss how the electronic properties of aromatic compounds determine the regioselectivity of cross-couplings. Unless there are other factors that override electronics and perturb innate reactivity, such as strong steric influences or directing groups, electronic effects will usually guide the reaction selectivity. For six-membered aromatic systems, electronic effects from present substituents are the main factors controlling regioselectivity. To understand the electronic effects caused by electron-donating or electron-withdrawing groups, you should review your knowledge in organic chemistry of arones. Particularly, electron-donating groups make the ortho and para positions relatively electron-rich. In such a system, if you have a halogen atom at the meta position, then the electron-donating group will drive the selectivity of cross-coupling to the meta position. For electron-withdrawing groups, the selectivity is reversed, as they make the ortho and para positions relatively more electron-deficient. As a result, electron-withdrawing groups direct oxidative addition to the ortho and para positions. In the case of aromatic heterocycles, the electronic properties of the system are controlled by the heteroatom, and as a rule, the most electron-deficient positions are alpha positions to the heteroatom. Let me show you two examples to make everything more obvious. One example of substituent effects on electronics and, by extension, guiding regioselectivity was described by Hearn and co-workers in their syntheses of unsymmetrical paraterphenyl moieties, which have been shown to possess medicinally relevant biological properties. This transformation was achieved through a sequential ligand-free Suzuki coupling of 1,4-dibromo-nitrobenzene and diverse aryl boronic acids with a palladium acetate pre-catalyst. Due to the electron-withdrawing nitro substituent, the first oxidative addition occurred preferentially at the more electrophilic carbon-bromine bond ortho to the nitro group, resulting in the ortho-substituted biaryl intermediate. Subsequently, this intermediate underwent a second coupling at the meta position upon the addition of the second structurally different aryl boronic acid to provide the desired terphenyl scaffold. Pyridines are inherently pi-deficient by virtue of the embedded electronegative nitrogen atom, which inductively withdraws electron density from positions C2 and C4. As a result, for cross-couplings of polyhalogenated pyridines, oxidative addition is also favored at the C2 and C4 positions, but this selectivity also greatly depends on specific reaction conditions, other substituents, and substitution patterns. In this example, you can see how selectivity can be achieved between the electronically similar C2 and C4 positions of 2,4 dichloropyridines by altering the substituent at the C3 position. When the substituent is equal to hydrogen, the intrinsic reactivity of this pyridine under still coupling conditions favors cross-coupling at the C2 position. Adding an electron-donating amino group to the C3 position also favors coupling at the C2 position, presumably due to the deactivation of the C4 position. However, adding an electron-withdrawing nitro group to C3 activates the C4 position toward oxidative addition, thus completely reversing the selectivity of the reaction. For aromatic systems containing two or more of the same type of halogen, several indicators based on experimental data have been identified to help predict the intrinsically most reactive positions for oxidative addition. For instance, analysis of the proton NMR chemical shift values of the parent non-halogenated arones can serve as a guide for predicting the site of cross-coupling reactions. The position of the most deshielded proton when substituted by a halogen will be the favored site for the first oxidative addition. Remember, the higher the chemical shift of the proton, the more electron-deficient is the carbon bound to the proton. Here are presented two examples as proof for the concept. Proton NMR of the parent non-halogenated pyrrole suggests that for this 2,3 dibromopyrrole, the most reactive site should be position 2. This was proved to be true for the Suzuki coupling reaction presented here. For the second example, similar NMR studies proposed that position 3 should be more reactive than position 4, which was confirmed by a control Suzuki coupling. Here, more examples are presented for the use of NMR prediction of the regioselectivities for 5-membered, 6-membered, infused heterocycles. The numbers show the proton NMR chemical shift values of the parent non-halogenated arones. Based on these numbers, the positions indicated in blue should be the sites for the first oxidative addition. The positions indicated in red are the second favored positions for corresponding couplings. The positions in green are the most electron-rich positions and are the least favored sites for oxidative addition. Whereas the relative carbon electrophilicity in an aromatic ring play a prominent role in oxidative addition selectivity, one must also consider the steric environment and its effect on regioselectivity. In cross-coupling, selectivity is often governed by the interplay between electronics and sterics, with the most electrophilic and most sterically accessible position being primed for oxidative addition. Among others, sterics can differentiate between two electronically similar positions, and steric effects can reinforce the selectivity dictated by innate electronics. One example of steric effects overriding electronic effects is underlined in red, where coupling takes place at the more sterically accessible C5 position of 2,5 dibromofuran. Despite the C2 position being electronically favored by the virtue of being adjacent to both an oxygen atom and an electron-withdrawing ester moiety. In the case of 2,5 dibromothiophenone underlined in blue, there are no pronounced electronic effects from the pendant tetrahydroisoquinoline derivative. Therefore, the C2 and C5 positions are electronically similar, and in this case, the difference in the steric environments between these two positions facilitates reactivity at the more sterically accessible C5 position. The C2 positions of thiols underlined in green are both electronically preferred over the C5 and C3 positions and are more sterically accessible. Therefore, sterics reinforce the innate electronics of these systems, and coupling occurs at the C2 position in both cases. The extra-functional groups present in polyhalogenated substrates, among others, can have coordinating ability to the catalyst and can act like a directing group, guiding the catalyst to a proximal carbon heteroatom bond, thus enabling regioselective oxidative addition and subsequent functionalization. The functional groups on the polyhalogenated substrate can also coordinate to the ligand of the catalyst and guide the catalyst to the position for oxidative addition through the ligand of the catalyst. The transition metal binding directing groups are optimal for proximal carbon heteroatom bond cleavage, whereas ligand binding directing groups can be applicable for remote carbon heteroatom bond cleavage. Let me show you a few examples for clarity. Basic nitrogen-containing heterocycles are a common class of transition metal-binding directing groups for the selective functionalization of polyhaloarenes. For instance, nickel-catalyzed Negishi coupling, including perfluorinated arenes, can be made regioselective by the application of directing groups based on pyridine, quinoxalin, and oxazolene. The ligand-binding directing group strategy is particularly useful for substrates possessing functional groups that cannot act as transition metal-binding directing groups. For instance, phenols are not particularly good ligands for late transition metals. However, they are capable of forming heteroaggregates with specialized phosphine ligands possessing a hydroxyl group, as presented here. These non-covalent interactions can place the palladium catalyst in close proximity to the carbon-halogen bond in the ortho position of phenol, thus enabling regioselective Sonogashira coupling followed by cyclization. Certain ligands that have adequate length and can bind to a functional group present in the substrate serve as a linker between the substrate and the transition metal complex, enabling the catalyst to come in reasonable proximity to the remote carbon-halogen bond for activation. Non-covalent interactions involving sulfonylated phosphines in the amino group of polychlorinated systems, as presented here, can be applied for the regioselective Suzuki coupling at remote positions. The regioselectivity of the reaction can be controlled by the position of the sulfonyl group. The best selectivity for the meta-arylation product was obtained when using the sulfonylated derivative of S-phos combined with palladium acetate. Another factor affecting selectivity was the substituent at the amino group. More polar substituents like triflate enhance the electrostatic interactions between the ligand and the amino group, improving the meta-selectivity of the reaction. Reactions performed with non-sulfonylated ligands resulted in a statistical mixture of regioisomers, supporting the proposed hypothesis on the control of regioselectivity by non-covalent interactions. The majority of examples for the site-selective cross-coupling of polyhalogenated arenes discussed so far have leveraged key differences in reactivity between various carbon heteroatom bonds and the directing abilities of functional groups present on the substrate. Alternatively, ligands that bind exclusively to the transition metal can be used to tune the electronic and steric properties of the catalyst, thus controlling the regioselectivity of a given cross-coupling involving polyhalogenated substrates. In other words, the right ligand-transition metal combination can define the regioselectivity of cross-couplings regardless of the steric and electronic nature of the used starting materials. The reasons controlling the regioselectivity enabled by a certain ligand are not always clear. Here is a good example describing a ligand-controlled site-selective Suzuki coupling of 3,5-dichloropyridazine. The reaction can produce a mixture of three products, including the biaryl formed by the arylation at position C3, the product from arylation at position C5, and diaryl pyridazine produced by double arylation. Based on the electronic properties of the system, 3,5-dichloropyridazine is expected to react more readily at position C3, as the bond dissociation energy of the carbon-chlorine bond alpha to the nitrogen atom is lower. Screening a wide range of ligands indicated that electron-deficient bidentate ligands and triphenylphosphine favor the formation of the C3 coupled product. The best yield and selectivity were achieved using the DPPF ligand. Notably, an electron-rich analog of DPPF gave the opposite selectivity, albeit in a moderate ratio between mono and bis-coupled products. This prompted the authors to screen more electron-rich monodentate phosphines. As a result, it was found that Buchwald's biaryl monophosphine ligands predominantly favor the formation of the bis-coupled product, while Q-phos gave the best selectivity for the C5 coupled product. It was hypothesized that electron-deficient ligands favor slower oxidative addition. Therefore, the insertion of the metal occurs at the more reactive C3 position, as predicted by the electronic properties of pyridazine. On the contrary, bulky electron-rich ligands direct the metal center to insert into the more accessible C5 position, as the lone pairs at nitrogens form a nucleophilic pool shielding the position C3 for the electron-rich catalytic system. Altering the used ligand is not the only factor that can potentially control the regioselectivity of cross-couplings. The first example of shifting the regioselectivity by modifying only the reaction solvent was disclosed in 2008. In a Suzuki coupling between dibromopyrrole and 4-methoxyphenylboronic acid, in a 3-to-1 mixture of toluene and ethanol, a mixture of isomer arylation products was obtained in a ratio of 22-to-1, with the C5 coupled product isolated in 70% yield. Alternatively, when the reaction solvent was changed to DMF, the C3 coupled product was obtained in 53% yield. In general, it was found that under otherwise identical conditions, more polar solvents such as DMF and DMSO favor C3 coupling, whereas relatively less polar solvents favor C5 coupling. NMR studies indicated that the electronics of C3 and C5 positions changed depending on the differential solvation. In nonpolar solvents, the difference in proton NMR chemical shifts between the C3 and C5 protons was barely detectable, while in DMSO, the difference between the C3 and C5 protons was more pronounced, with the C3 proton remaining more deshielded. Given that oxidative addition is generally favored at the more electron-deficient positions, these NMR studies were in good agreement with the experimentally observed selectivity trends. In a related study on the Suzuki coupling involving 3,5-dibromopyrrone, it was found that in nonpolar solvents, the reaction delivers the C3 coupled product. On the contrary, in more polar solvents, such as DMSO and DMF, and in the presence of copper iodide, the C5 coupled product was generally formed as the major product. Notably, the C5 coupled product was observed only in the presence of copper iodide, and conducting the reaction in the absence of copper iodide exclusively generated the C3 coupled product. Control experiments on the oxidative addition of palladium to 3,5-dibromopyrrone were consistent with the Suzuki coupling results. The C3 palladium complex was formed in nonpolar solvents like toluene, irrespective of whether copper iodide was added. In polar solvents like DMF, the C5 palladium complex was formed only in the presence of copper iodide. Computational studies indicate that copper iodide assists in the dissociation of phosphine ligands from palladium, leading to monoligated palladium species, which are more reactive and can initiate, among other things, oxidative addition at the less reactive C5 position. Chemoselective reactions, which discriminate between electrophiles such as organometallics, are well established. In contrast, the development of nucleophile selective reactions has seen limited success. Nevertheless, modern discoveries in the field of organoboron chemistry provide us with effective tools that can enable site-selective coupling in systems possessing multiple carbon-boron bonds. As you may remember, for transmetallation involving organoboron reagents, one needs to use a base to activate the boron. Boronic acids and their esters have an empty p orbital at the boron and behave like Lewis acids. The applied base coordinates to the empty p orbital of boron, increasing the nucleophilicity of the system and facilitating transmetallation. The key for a successful boron-selective reaction is to discriminate between the boronyl groups that are exposed to the same reaction conditions. Appropriate use of masking groups on the boron atoms can play a critical role in controlling their reactivity. So far, two efficient masking groups were introduced for boronic acids. One is based on 1,8-diaminonaphthalene and is abbreviated like DAN. The other is based on N-methylamino-diacetic acid and is abbreviated like META. These masking groups reduce the Lewis acidity of boron by delocalization of electrons from lone pairs of nitrogens to the empty p orbital of the boron. This, in turn, protects the boron from reactions involving bases and prevents transmetallation. DAN and META masking groups are stable under basic conditions, which are typical for most of the cross-couplings. On top of that, these masking groups can be removed at any point by acidic hydrolysis, generating free boronic acids that can be applied for later functionalization. Here is an example of chemoselective Suzuki couplings involving multiple organoboron reagents. Treatment of an aryl boronic acid with an aryl bromide possessing boron masked with a diaminonaphthalene group will lead to a single product where the boron masked with diaminonaphthalene will stay intact. Later, the diaminonaphthalene protecting group can be removed by acidic hydrolysis, and the formed boronic acid can undergo another Suzuki coupling to deliver this terphenyline derivative. This approach has some similarities to peptide synthesis and is ideally suited for manual or automated generation of diverse polyurenes. Here is an example covering the N-methylamino-diacetic acid masking group. N-methylimino-diacetic acid-protected aryl boronic acid can undergo a selective CH borylation catalyzed by iridium. The newly introduced pinocol ester of boronic acid can be consumed in a consecutive Suzuki coupling without affecting the masked boron. In summary, while the chemoselective functionalization of a carbon-halogen bond in a molecule bearing multiple distinct halogens can be quite straightforward, predicting and rationalizing the selectivity outcome in an arene bearing multiple, but identical halogen atoms has remained challenging. Therefore, several factors have been identified to enable differentiation between carbon positions bearing identical halogen atoms. Initially, this distinction was achieved purely by leveraging the inherent electronics of the system, where the most electrophilic carbon-halogen bond would undergo faster oxidative addition. In addition to electronically driven regioselective cross-coupling processes, steric effects were also identified to guide oxidative addition to the least sterically encumbered carbon position. Following these advances, the coordinating ability of functional groups present in the substrate was leveraged to direct oxidative addition to a particular position. Subsequently, methods were developed to achieve regioselectivity by tuning the electronic and steric influences of an external ligand. More recently, selectivity has been achieved by altering the choice of reaction solvents and additives. The following lecture will demonstrate how the knowledge you have gained up to this point can be applied in the production of value-added products. Thank you for your attention.